Good morning. I would like to start by acknowledging the invitation to be here. And I'll speak about a neglected disease that has been called by many as the mysterious disease. So I'm coming from the biomedical field, and I'll try to show you today is what we know about the host-parasite interaction in this infection and what we know about transmission. But the main point is that there are many questions to be answered. And this is one of the cases of the neglected diseases in which research is really important. So the disease was described not more than 60 or almost 60 years ago. Is a recent disease caused by Mycobacterium ulcerans that causes a necrotizing infection in the skin. Being a recent disease, it is already the third mycobacterial infection at the global level in tropical and subtropical areas, mainly in rural settings, and is particularly important in Africa, in Central and the West Coast of Africa, and also in Australia. So what do we know about this disease? It usually starts as a non-ulcerative skin lesion that then can evolve to the typical ulcer that we call Buruli ulcer. In addition to the clinical diagnosis, it is important to confirm in the laboratory that indeed it's an infection by Mycobacterium ulcerans. And the most important laboratorial test is a PCR for an insertion sequence that is very rich in the genome of Mycobacterium ulcerans. You'll see later that this is also important for tracking the bacteria in the environment. We don't have a specific vaccine against the disease, and the treatment sometimes is difficult. So now the WHO recommends an antibiotic regimen with streptomycin and rifampicin, which is quite effective in early lesions, but not in advanced lesions. And in these cases, surgery is needed and is associated with high morbidity, such like scarring, retractions, and even sometimes amputation of limbs. What do we know about the pathogen and about the host? So Mycobacterium ulcerans is a slow-growing bacteria. It takes up to 2 months to grow in culture media, and is genetically very closely related with Mycobacterium tuberculosis and Mycobacterium marinum. But interestingly, the optimal growth temperature is lower, around 30 degrees, which explains, at least in part, the preference of this bacteria for the skin. In a very peculiar way for a Mycobacterium, M. ulcerans secretes a very potent toxin, mycolactone, which was shown to be very important as a key pathogenic factor, both in vitro models and in vivo models, since when we inject the toxin in the skin of the guinea pig, we are able to mimic the infection by the virulent bacteria. And what do we know about the host? There are many questions about the host immune response, about transmission, but I'll try to focus my presentation first, very briefly, on host-parasite interactions and then in the transmission. So when we started working with this disease some 10 years ago, the description in the literature of the lesions in patients was a huge area of necrosis, as seen here in this skin section, with the erosion of the epidermis and the formation of an ulcer. And Mycobacterium ulcerans, which is here stained in red, was described as an extracellular pathogen in the middle of necrotic tissue. So we started by looking to this disease and trying to figure out if immune cells like macrophages and lymphocytes were indeed absent in this infection or if they are recruited to the infection site. So we have used mainly the mouse model of infection in which you can see edema and ulceration 4 weeks after infection with a virulent strain of Mycobacterium ulcerans, but also studying in vitro models and samples from the patient. Very briefly, what we found is that indeed very early after infection, Mycobacterium ulcerans induces an immune response with the recruitment of cells, namely phagocytic cells such as the macrophage, and that M. ulcerans is able to grow inside the host infected cells. And by the secretion of mycolactone, is able to induce the lysis of the infected macrophage, and this is the image that prevails in advanced human lesions, a huge area of extracellular bacilli, but at the periphery we found that are always present inflammatory cells, namely phagocytic cells. So this is not the main topic of my presentation for this session here, But I would like to tell you that this has implications for the control of the disease. Namely, because showing that we have an intermacrophage growth phase, this gives important information about what type of vaccine should be developed. Namely, we know that cytokines like gamma interferon, which are produced by T cells, should be one of the targets for a vaccine. This is also important for treatment, because it is important to know that we should use antibiotics that are able to cross the macrophage membrane and to target the intracellular bacteria. So this is one of the main areas of research in our group. But if this is important to control the infection, if you need to develop a vaccine or to know what kind of antibiotics should be used, transmission is also very important. So what do we know about water and the transmission of this disease? It is clear that there is a strong association between flooded areas and swamps and the disease. This was proved in areas after heavy rainfall, but also in environments that are disturbed by human activity, namely related with agriculture, such as damming, deforestation, construction of irrigation systems, as well as other human activities like mining activities, and also expansion of populations to areas close to water bodies, usually stagnant waters or slow-flowing waters. So this is strong data showing that the outbreaks of the disease are related with these type of aquatic environments after human disturbance. But we should be able to track the bacteria in these environments. So what has been used by many researchers is the same PCR that amplifies part of the genome of the bacteria for the diagnosis in patients, and then to use these PCR techniques in samples from the environment, namely from the aquatic environment. And what was found, both in Africa and in Australia, was that the message for this PCR was positive in many organisms of the trophic web of the aquatic environment, not only in detritus and in biofilms of plants, but also in insects and invertebrates such as amphibians and fish. Can we prove that hemostasis is present in these animals of the trophic web? There are problems. First, this technique detects, the PCR detects the DNA from the bacteria, but not the whole microorganism or a viable microorganism. So one could argue that the presence of DNA could be just by washing into the aquatic environments of DNA from destroyed bacteria. The second problem is that also other mycobacterial species that are present in the water also have this sequence insertion. So there is a problem about relating the presence of the DNA of the bacteria and the transmission of the disease. In addition, there's not clear the association between the rivers and the swamps who have a higher message for this PCR and the outbreaks of the disease. But again, there is knowledge that is absent. We don't know which is the time of incubation of the disease. It can vary from 2 weeks to 7 months. So it's very difficult to link the presence in a specific area of the PCR positive message and the disease. So what we have done a few years ago, taking in mind this information, and also some experiments done by the laboratory of Laurent Marsulier. And Laurent Marsulier has done a very clever experiment. So he infected in the lab aquatic bugs with hemostrans by feeding these bugs with contaminated prey, and then mice were bitten by these bugs, and he was able to transmit to the mouse in the laboratory the disease through aquatic insects with the clinical features of Buruli Ulcer. So our hypothesis was that hemostrans was indeed present in the environment as a viable pathogenic agent and maybe in insects. So what we have done was to study a high number of aquatic insects from Benin, and we have prepared homogenates from these insects, and we have cultivated these homogenates after decontamination in rich bacterial media, tracking always for the PCR message. So after some passages in vitro, we injected these homogenates in the mouse. And when disease was present, we have done successive passage of these homogenates. And after this work, for one of the insects we have tried, the Gerridae, it is a water strider, we could find in the mouse the typical signs of the disease. It was possible to isolate in a pure culture Mycobacterium ulcerans that we characterized genotypically and phenotypically, and we proved that this species was producing mycolactone and that the pathology in the mouse was the same as the human pathology. So what this data shows is that indeed Mycobacterium ulcerans is present in aquatic environments as a viable pathogen. However, it does not prove that this insect is the vector of the disease, because usually, water striders do not bite humans, and they are not hematophagous. But this reinforces, that M. ulcerans is really an environmental pathogen related with water. Other type of work that we have been done as others is trying to identify new foci of the disease. Just as an example, this was a collaboration with Françoise Portals, as the previous work, and with Anatole Kibadi, and the idea was to look in the north of Angola for the presence of Buruli Ulcer. And why? Because you knew that in the Coango River basin, the activities of mining, of gold mining, were important and were done in aquatic environments. And also that in the Democratic Republic of Congo, the disease was endemic. So what we have done is to follow patients coming from Angola to Kinshasa that were for some months with a skin ulceration, that were huge lesions, as you can see in here, confirmed to be Buruli ulcer in the laboratory, that were cured by the WHO protocol of antibiotic treatment combined with surgery. But just pay attention to the number of months of hospitalization. So this points out how dreadful the disease is, even when we can treat the disease. So what we showed here is that indeed there are new foci of the disease that we don't know, namely the ones related with aquatic environments, such as the ones relating with mining activities. And also that the protocol that is recommended by the WHO is effective even for big lesions when we combine antibiotic therapy with surgery. So, but what are indeed the risk factors for Buruli ulcer? There are different studies looking to different aspects of the water access and water contact and trying to identify the risk factors. I would not ask you to read all these, because many of these findings are not consistent among different works. Maybe because of sampling procedures, maybe because of the different seasons of the year in which these samples were collected. What we can say for sure from the literature is that in addition to proximity to stagnant waters, risk factors include exposed skin, improper wound care, and in Australia, contact with mosquitoes. And avoiding these risk factors, populations are protected. There are some other information, but not solid information about other activities, like agricultural activities, aquatic-related activities, but the data is really not solid. What we know for sure is that in Australia, there are positive messages for the DNA of the pathogen in many aquatic samples, but also in mosquitoes. It is possible by PCR to amplify the genome of the pathogen in the salivary glands of the mosquito, and there are anecdotal descriptions of people being bitten by a mosquito and then developing a Buruli ulcer. Interestingly, in Australia, there are many cases, well-documented cases of infection and disease in mammals, both in wild animals like the koala and the possum, as well as in domestic animals. The picture is quite different in Africa. We also have the positive message in many types of aquatic samples, but there are no reports on infected mammals, both in the wild and in the domestic setting. So the fact is that we don't really know for sure which is the reservoir of the pathogen, and we don't really know how is the transmission. What we think is that, according to the data, in Australia the transmission can occur directly with skin contact from contaminated sources, like water irrigation systems. But we also know that wild mammals are infected, they harbor viable M. ulcerans bacilli that then could be transmitted to the mosquito, that then could be a vector for human disease. But until now, no one was capable of isolating Mycobacterium ulcerans in a pure culture from the mosquito, because many of these samples from the environment are also contaminated with other mycobacterial species that grow much faster than Mycobacterium ulcerans. So we are not sure. In Africa, we know that we have a positive message in the aquatic environment, in the trophic chain, namely in biofilms in plants. We also found a positive message in insects that are carnivorous insects belonging to the Bellostomidae family that can really beat humans. But they are not hematophagous, so this will be an accidental event and not really an event that will happen often. At the same time, the message for the PCR was found in many animals, both vertebrates and invertebrates, that then could be prey of these bigger insects, that then could be eaten by bigger fish or frogs, and both these sources could lead to infection of birds that could transmit the disease for other geographical areas around. So there are many questions, and the mode of transmission is not clear, but we know that there is a strong association with water, and maybe we can learn from you, from people working in other infectious diseases that are transmitted by water or related with water. Therefore, the control measures are difficult to define. What we know is that it is important to avoid wetlands and stagnant water sources in the endemic areas that are usually rural areas, and that is very important to wear protective clothing and to take care of wounds by cleaning these wounds immediately. So the control of the disease has been focusing in public awareness, in healthcare education, leading to detection of early lesions and early treatment. The good news is that the number of patients infected every year is under control and is even decreasing, as shown in here for Africa, which may point out for a good result of these strategies for control of the disease. But the main point is that really we don't know how the transmission occurs. Even we have different hypotheses for Africa and for Australia. Thank you very much.